Good day, everyone. Today, I will present our laboratory activity entitled Kinetics of Salivary Amylase, Effects of Temperature, pH, and Substrate Concentration. Salivary amylase is an enzyme found in saliva that begins the digestion of carbohydrates in the mouth by breaking down starch into simpler sugars. Since enzymes are sensitive to environmental conditions, their activity can change depending on temperature, pH, and substrate concentration. In this experiment, iodine solution is used to determine if starch is still present. A blue-black color indicates that starch is present, while the absence of the blue-black color means the starch has already been digested by salivary amylase. The objectives of this experiment are to measure salivary amylase activity through starch digestion time. Second, to determine how temperature and pH affect enzyme activity. Third, to analyze the effects of substrate concentration reaction rate. And fourth, to relate the findings to digestive physiology. The materials used in this experiment include filtered fresh saliva sample, 1% starch solution and other starch concentrations, iodine solution or IKI, buffer solutions at pH 4, pH 7, and pH 9, test tubes and test tube rack, pipettes or droppers, stopwatch and thermometer, ice bath at 5 degrees Celsius, room temperature set up at 25 degrees Celsius, warm water bath at 37 degrees Celsius, and hot water bath at 60 degrees Celsius. Before starting the experiment, the saliva sample was filtered through gauze to remove solid particles. Iodine solution was also prepared on a weight surface for easier observation of color changes. For the first experiment, which is the effect of temperature, we tested how temperature affects the activity of salivary amylase. 4 test tubes were labeled according to their temperature conditions, which are test tube A for 5 degrees Celsius, test tube B for 25 degrees Celsius, test tube C for 37 degrees Celsius, and test tube D for 60 degrees Celsius. Each tube received 5 ml of 1% starch solution and 1 ml of filtered saliva. The solutions were mixed gently and placed in their assigned temperature conditions for 5 minutes. After incubation, timing began immediately. Every 30 seconds, 1 drop from each tube was placed onto iodine solution to check if starch was still present. A blue-black color is indicated that starch was still present, while no color change indicated complete starch digestion. The time when the blue-black color disappeared was recorded as the starch digestion time. Among all temperature conditions, the test tube at 37 degrees Celsius showed the fastest starch digestion, indicating the highest enzyme activity. At 5 degrees Celsius, digestion was much slower because low temperature reduces molecular movement and enzyme-substrate collisions. At 60 degrees Celsius, the enzyme activity decreased because the enzyme became denatured. Denaturation changes the shape of the enzyme's active site, preventing proper binding with starch. This result shows that salivary amylase works best near normal body temperature. For the second experiment, which is the effect of pH, we investigated how pH affects salivary amylase activity. 3 test tubes were labeled Tube A for pH 4, Tube B for pH 7, and Tube C for pH 9. Each tube contained 5 ml starch solution, 1 ml filtered saliva, and 2 ml buffer solution of the assigned pH. All tubes were placed in a 37 degrees water bath to maintain a constant temperature. Every 30 seconds, drops from each mixture were tested with iodine until the blue-black color disappeared. The digestion times were then recorded. The results showed that salivary amylase was most active at pH 7, which is near neutral pH. At pH 4, enzyme activity decreased significantly because acidic conditions interfere with the enzyme structure and active site. At pH 9, activity was also decreased because highly alkaline conditions affect enzyme stability. This explains why salivary amylase works efficiently in the mouth but becomes less active in the acidic environment of the stomach. For the third experiment, we studied the effect of substrate concentration on enzyme activity. 4 test tubes were prepared with different starch concentrations, which are 0.5%, 1%, 2%, and 3%. Equal amounts of saliva were added to each tube, and all tubes were maintained at 37 degrees Celsius. The stopwatch was started immediately after mixing. Every 30 seconds, drops from each tube were tested with iodine to monitor starch digestion. The time needed for the disappearance of the blue-black color was recorded. As starch concentration increased, the reaction rate also increased because more substrate molecules were available for enzyme binding. However, at higher concentrations, the increase in reaction rate becomes less noticeable because the enzyme's active sites became saturated. This demonstrates the concept of enzyme saturation, where all available active sites are occupied and additional substrate can no longer significantly increase the reaction rate. This experiment demonstrates the importance of optimum conditions for enzyme activity. Salivary amylase functions best near neutral pH because the oral cavity normally maintains a pH close to 7. Under acidic conditions, such as in the stomach, the enzyme becomes less active, slowing starch digestion. At very high temperatures, enzymes undergo denaturation. This changes the three-dimensional structure of the enzyme and destroys the active site necessary for enzyme substrate binding. Substrate concentration affects reaction rate because increasing substrate increases enzyme substrate collisions. However, once saturation occurs, the reaction rate levels off. If the saliva sample were diluted, enzyme concentration would decrease, leading to a slower digestion of starch and longer digestion times. One limitation of this experiment is that enzyme activity was measured through disappearance of iodine color rather than using a spectrophotometer, which would provide more precise quantitative measurements. Possible sources of error includes inconsistent timing intervals, unequal saliva volumes, improper temperature maintenance, and contamination of samples. In conclusion, this experiment successfully demonstrated how temperature, pH, and substrate concentration affect the activity of salivary amylase. The enzyme showed optimal activity at approximately 37 degrees Celsius and pH 7, which are conditions similar to the normal environment of the human mouth. Extreme temperatures and non-neutral pH levels reduced enzyme activity because they alter the enzyme structure. Increasing substrate concentration increased the reaction rate until enzyme saturation occurred. Overall, this experiment highlights the importance of enzyme kinetics in digestion and explains how environmental conditions influence influence physiological processes in the human body. Thank you for watching this laboratory presentation.